we are in proteins. Okay,
Yes, ma'am.
proteins are used for bodybuilding. All major functional structural aspects of body are carried out by protein molecule. Abnormality in protein structure will lead to molecular diseases with alterations in metabolic functions. Then what are the contents of protein? You know, they are
Yeah.
formed of
Carbon.
amino acids.
Yes, yes,
Yes.
ma'am. Yes, ma'am.
So the components are carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. Okay. Sometime in case of phosphoprotein, milk protein, casein is a phosphoprotein that contain phosphorus.
Yes, ma'am.
Yes.
Yes, ma'am.
Then sulfur containing amino acid is there. So proteins can contain sulfur.
Sulfur also, yeah.
Also. Then nitrogen is the main characteristic of protein, and the nitrogen content of ordinary protein is 16% by weight. It's a multiple choice questions. Last years there are multiple choice questions for BDS and MBBS. Here now MBBS for multiple MCQs are there. So nitrogen content of ordinary protein is 16% by weight. Okay.
Okay, ma'am.
What is your idea about peptide bond? What is meant by peptide bond?
Ma'am, there
Mm.
peptide bonds are there in
Mm.
this thing, no? We studied that, right?
Mm.
Peptide bond is a bond between in tertiary and in the like folds of proteins, right?
Mm. What are proteins? What are proteins?
Ma'am, amino acids, a chain
Proteins are
of
made up of amino acids.
amino.
Okay,
Yeah.
so
Yes, ma'am.
there is only one amino acid, it is known as an amino acid. Okay, if
Yeah.
there are two amino acids, these two amino acids are linked together by a special bond.
That is peptide bond,
Okay,
right?
that is known as peptide bond. Got
That's
the
why it is
point?
folds of proteins, right?
Yes, folds of protein.
Yeah, in
Okay. Okay, okay.
tertiary and
So
quaternary there are folds, right, ma'am?
yes, yes. Okay, okay. But the basic definition of peptide bond is alpha carboxyl group of an amino acid reacts with alpha amino group of another amino acid to form a peptide bond. Okay. Did
Okay,
you
ma'am.
get the point? You know this general structure of amino acid?
General structure of amino acid.
What is amino acid? Actually, the structure. What is amino acid?
There are four structures, right, ma'am?
Four structure. Amino acid contain two functional group. Which are the two functional group?
This
Amino
C
group of
C no, that's not
yes,
it.
yes. You
CH,
tell me.
yeah.
Don't worry. You can tell me.
Is it no,
Ah,
that's carbo.
you don't worry.
Amino acid. Amino
Hmm,
acid is
hmm. Amino acid
mm,
we can express as NH2 CH
okay.
COOH R.
Yeah.
Okay.
Yes, ma'am.
CH in
Yes,
the
ma'am.
middle. We can write it as NH2 and COH in both sides because amino acid, amino
Yeah.
group and carboxyl group. COOH represents carboxyl group. Right,
Okay.
Vishnu? There
Yes,
is
ma'am.
a side chain R. Based on the side chain, we have 20 different amino acids.
Okay, ma'am.
Clear?
Yes,
Yes.
ma'am.
So here alpha carboxyl group of one amino acid that reacts with alpha amino group of another amino acid forms a peptide bond.
Hmm.
Okay.
Yes,
So
ma'am.
one amino acid is there. It is known as amino acid. There are two amino acid. It is known as dipeptide.
Okay,
Okay.
ma'am. Okay,
How
ma'am.
many peptide bonds are there in dipeptide? One.
Two. I mean, oh yeah, two amino acids. One. Yes, ma'am. Yes, ma'am.
Then what about in tripeptide?
Three. No, two.
Two
Two.
peptide bonds and three amino acids. Okay, then what is meant by an oligopeptide?
Four or more, right?
Uh, Four or more. Oligosaccharide in case of carbohydrate, oligopeptide in case of protein that contain 3 to 10 residues.
Okay, ma'am.
Okay, 3 to
Hmm.
10 residues. Then 10 to 50 amino acids are called as polypeptide.
Yes, ma'am.
Okay,
Hmm.
more than 50 amino acids are called proteins.
Protein.
Okay, then here is the slide for peptide bond formation. You can see the structure here. Yes.
Yes, ma'am.
Okay, carbon valency is 4. It is satisfied by different groups. You can see CHR, NH3 plus and COH. These are the groups attached to it. Okay, so carboxyl group of one amino acid and amino group of another amino acid join together and one molecule of water is removed, this peptide bond is formed. C double bond O NH. Yes, Vishnu? Is it clear?
Yeah.
So how can we express this peptide bond? What is the definition of peptide bond?
It's a bond between two amino acids that are formed when COH group and NH3 group
COH
join
group
together.
of one amino acid.
Sorry, one
Should be
amino acid.
clear.
And
Okay. And amino group of another amino acid.
Join together and release H2O water molecule out.
Then what is the structure of peptide bond? C double bond O NH.
Yes,
okay. Is it cis or trans? The presentation is cis or trans in nature?
Trans, right?
Yes, very good. Trans in nature. Okay. So proteins have different level of structural organization. We know it: primary, secondary, tertiary, and quaternary. So primary structure that explains the sequence of amino acid in structure. It is the most simple structure, basic structure. Okay.
Yes,
so primary structure denotes the number and sequence of amino acid in the proteins. The higher level of organizations are decided by this primary structure. Okay. So primary structure is maintained by covalent bonds of peptide linkage. You can write like glycine, alanine, valine. Okay.
Yes, ma'am.
It's a peptide. Then glycine, valine, alanine. What is the difference? Both contain 3 amino acids, same amino acid. Is it same?
No, it's
Vishnu?
not same, no.
Why? Why? Both
Because
the tripeptides
order is
contain
different.
same amino acid. What is the difference?
Order is different.
Yes, their sequence is different. Okay. In case of replication, transcription, translation, we know there is a single amino acid change will cause a mutation. Have you heard about mutation? Vishnu?
Yes, ma'am.
What is meant by mutation? Sudden genetic change.
It's a change in the DNA. Yes, ma'am.
Okay. If there is a change in amino acid, that will create another protein. Okay.
Yes,
so in this tripeptide, why it is known as tripeptide? There are 3 amino acids. Okay. How many peptide bonds are there? 2.
2.
Okay. But it is different. These first and second peptides are different because their sequence is different. Okay.
Yes,
then the peptide bond is partial. What are the characteristics of peptide bond? Peptide bond is partial double bond in character. C and bond, that is the peptide bond. It is trans in nature. There is no freedom of rotation because of this partial double bond character. Okay. The distance is 1.32 angstrom, which is a midway between single bond and double bond. Then there is a particular term. We are familiar at the time of entrance. You know, Ramachandran ankle is there. A special type of angle of rotation. Okay, in peptide bond, it is known as Ramachandran angle that determines the spatial orientation of the peptide chain. Then next is about the numbering of amino acid in protein. It is known as N-terminal to C-terminal. Yes? What is this? N-terminal to C-terminal. What is this? Any idea?
That's amino group of one and the COH group of the other. Right?
You can see from this diagram, amino group of first amino acid is free. Okay? It is, you can see the diagram.
Yes, ma'am.
Yes. Here, these amino acids are linked together through peptide bonds, right? So, in case of first amino acid, this amino group is free. Okay? And the COH group is involved in peptide bond formation. Glycine, COH group is involved in peptide bond formation with amino group of isoleucine. Okay.
Yes, ma'am.
Then last amino acid is methionine. Okay. In the case of methionine, COH group cannot be involved in peptide bond formation. So we have one amino group free in the first amino acid and one COH group free in the last amino acid. Yes?
Yes, ma'am. Yes.
Got the point? That's why it is known as N-terminal to C-terminal.
Yes.
Okay. N-terminal. In all these proteins, we can see this term, N-terminal to C-terminal. You don't know what is it. One amino group is free in first amino acid, one COH group is free in last amino acid. Then how can we write in the name of peptides? Amino acid and polypeptides are named changing the suffix INE to YL. Okay. Glycine. When we write in the form of peptides, we can write like gamma glutamyl cystinyl glycine. Okay. This is a tripeptide that contains, which are the amino acid present in this tripeptide? Vishnu.
Cysteine is there, glutamine is there, and glycine.
Which is the first amino acid? Your sound is very low. What happened?
Hello, ma'am.
Yes. Glutamic acid.
Then cysteine.
Sleepy now?
No.
Not interested? No. Are you okay?
No. Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Then what is glutamic acid? Cysteine and glycine.
And glycine.
Okay.
Yes, ma'am.
So here, when we write the name of peptide, we convert INE to YN. Okay.
Yes, ma'am.
Okay.
Yes, ma'am.
You can see the different structure. Can you name it? Which are the different structure? First one is primary. Okay.
Primary, secondary, tertiary, and
secondary. Secondary, there are two types of structure in secondary. Which are they? You remember?
Secondary, two types. One is alpha and beta.
Alpha, helix, and beta pleated sheet structure. Okay.
Yes, ma'am.
Then next is the tertiary protein structure. In case of tertiary protein structure, what is the important? It's a three-dimensional representation.
Yes.
Okay. Just like this, twisting and folding and forms tertiary structure. What about quaternary protein structure? Different polypeptides are there. Okay. Each polypeptide, you can see one light color and dark color. Yes. So there are two polypeptide chains. Each polypeptide chain acts as one subunit. So the joining of subunit is there. Yes, that's about the quaternary structure. So primary structure that contain only peptide bond and sometimes disulfide linkages. Then secondary structure like alpha helix and beta pleated sheet that contain additional bonds like hydrogen bond, and so many bonds are there. Then tertiary structure, three-dimensional protein structure. Quaternary structure, polypeptides linked together. Okay. Okay, Vishnu? Yes. Okay. Then you can see the structure of insulin. Okay.
Okay, ma'am.
What is your idea about the structure of insulin?
The sulfide bond, insulin. Yeah, sulfide bond is there no in between them?
Okay, sulfide bond is there. How the sulfide bond forms?
Then
cysteine.
Formed between cysteine. Yeah.
Yes, cysteine contain a special group. Which is that special group?
Cysteine contain a special group.
We complete our sulfuration reaction. Yes. So that's the compound formed from cysteine.
Yes,
so which is the special group present in cysteine? SH.
SH. Yeah. Yes,
it is known as sulfhydryl group.
Yes.
Okay.
Yes.
That's why I told you.
I know.
Okay. Are you okay?
What happened was
Okay.
I was lying down and suddenly he told us class, so I jumped up and got it.
Okay. Cysteine that contains SH group.
Yes,
okay. That is sulfhydryl group. So when two sulfhydryl groups are linked together, removal of one molecule of hydrogen and it forms SS bond, disulfide linkage. Okay. You can see this interchain disulfide linkage and intra-chain disulfide linkage. Okay.
Yes, ma'am.
What is meant by interchain disulfide linkage?
Between A chain and B chain.
In between the chains. What is meant by intra-chain disulfide linkage? Within the chain.
Between the same chain. Yeah.
Okay. Within the chain. Okay. Between the same chains. Okay.
Yes.
Okay. These are also this is an example for our primary structure. Insulin structure is an example for our primary structure. Okay. Okay, Vishnu.
Yes, ma'am.
So here we complete our primary structure. Okay, these are the primary structure. And then next we are entering into secondary structure. Before entering into secondary structure, primary structure determines the activity. Okay?
Yes.
Activity of protein. A protein with a specific primary structure when put in solution will automatically form its natural three-dimensional shape. So the higher levels of organization are dependent on primary structure. Even a single amino acid change, we already discussed it, mutation in the linear sequence may have profound biological effects on mutation. For example, you know sickle cell anemia, HBS.
Yes,
What is
ma'am.
HBS? What is HBS?
Sickle cell anemia is because of the shape change in the uh, red blood cell, right? Where
Okay. What is
another
the
group
normal
is
shape of RBC? What is the normal shape of
concave,
RBC?
biconcave
Biconcave disc-shaped RBC that
yes,
changed into sickle shape. That's
shape.
only the structural difference. What about the chemical difference?
So the glutamic acid in the
Hmm.
RNA is
You can
replaced by
try to read
valine
from the
mutation.
slides. Okay. So
Huh?
hemoglobin that contain 4 polypeptide chain. What is the structure of normal hemoglobin HbA? Adult hemoglobin HbA that contain
For
alpha. Okay, 4
yes, ma'am.
polypeptide chains, alpha 2 and beta 2. Okay,
Yes, ma'am.
so
Yes, ma'am.
in the beta chain, okay, sickle cell anemia that occurs in the beta chain, the sixth position of amino acid is replaced by valine. Actually, the sixth position is glutamic acid. It is replaced by valine. We already discussed if there is a single change of amino acid that will create another protein, right?
Yes, ma'am. Yes,
So,
ma'am.
in case of hemoglobin, adult hemoglobin HbA that contain 2 alpha chains and 2 beta chains. Okay,
Yes,
what is the
ma'am.
difference between HbA and HbS? HbS means sickle cell hemoglobin. Okay, HbS, the sixth amino
Yes,
acid in
ma'am.
beta chain is actually glutamic acid. It is replaced by valine. Okay, so
Valine.
all the activities of protein that is based on this primary structure. Clear?
Yes, ma'am.
Yes.
Yes, ma'am.
Which are the other higher level of organization? Secondary, tertiary,
Tertiary and
quaternary.
quaternary.
Okay.
Yeah.
So next we are entering into secondary structure of protein. Okay. We have a
Yes, ma'am.
straight chain in case of primary structure. Okay. Now we are twisting
Yes,
or
ma'am.
folding. Okay. The conformation of polypeptide chain by twisting or folding is referred to as secondary structure. The amino acids are
Yes,
located
ma'am.
close to each other in their sequence. Yes, if it is in a
Yes, ma'am.
straight chain, they are too far. Okay. Hmm? Long
Yes, ma'am.
distance between two amino acids. But when they are twisted or folded, it is very close to each other in their sequence. Okay. Two types of
Yes, ma'am.
secondary structures are there: alpha helix and beta pleated sheet structure. One more structure we know that it is known as collagen triple helix. Okay, have you heard about
Yes,
collagen's
ma'am.
triple helix? Yes.
I've heard of collagen.
Collagen, the structure of collagen is triple helix. Okay. Mm.
Yes,
Now
ma'am.
you just understand it's a structure of collagen. The term secondary
Mhm.
structure denotes the configurational relationship between residues which are about 3 to 4 amino acids apart in the linear sequence. Okay.
Yes, ma'am.
There are different types of bones present in the protein structure. Which are the main bones present in the primary structure?
Which are the
Vishnu.
main bones?
Mm.
Bones as in them?
Bones means the
Oh,
linkage.
bones. It's inter peptide bonds, right?
Peptide bond, okay. One more bond is there.
Must be no,
We
not alpha.
discuss in the structure of insulin,
Ma'am, disulfide bond.
disulfide bond. Okay, these are the only two bonds present in primary structure. Which are they? Peptide bonds and disulfide bond. Okay,
Disulfide.
Vishnu.
Yes, ma'am.
Okay.
Yes,
In
ma'am.
addition to this peptide bonds and disulfide bond in the primary structure, secondary and tertiary levels of protein structure contains non-covalent bonds like hydrogen bonds, electrostatic bonds, hydrophobic interactions, and van der Waals forces. Okay. When
Yes,
we
ma'am.
twist or fold these amino acid structure, primary linear sequence of amino acids, there is a chance of non-covalent bonds, hydrogen bond, electrostatic bond, hydrophobic interactions, and van der Waals force of attraction. Okay,
Yes,
that hold all these amino acids together. Okay, secondary, you can see from the structure. This is linear structure, and this is the beta pleated sheet, and this one is alpha helix. Okay, then you can see this is known as alpha helix. Yes.
Yes, ma'am.
Yes, alpha helix. But now the amino acids are more closer. Yes.
Yes, ma'am.
Yes, all right.
Yes, ma'am.
Then you can see this NHC double bond O. Okay, NH. Then the special group is projected outside, that is R group. Then you can see the C double bond O is there. Then there is a connection between NH and this H of this NH bond and O of the CO bond.
Yes, ma'am.
Yes, that is known as hydrogen bond. Okay,
Yes,
so presence of hydrogen bond is there in secondary structure. Okay,
yes,
so now we are entering into the features of alpha helix. Okay, so alpha helix is the most common spiral structure of protein. It has a rigid arrangement of polypeptide chain. Alpha helix is tightly packed coiled structure with amino acids. Side chains are extending outward from the central axis. You can see from this diagram, the green color that represent the side chains. Okay?
Yes, ma'am. R group. Yes.
Yes, R group. In case of cysteine, which is that side group? Side chain.
In case of cysteine, it's CH2SH. Sulfur.
CH2SH. What about in serine? Serine and cysteine are almost the same. Serine, it is CH2OH.
Okay, wait.
Serine contain hydroxyl group. Cysteine contain sulfhydryl group.
SH, yes, ma'am.
Okay, yeah, so now you understand what is this R. Yes. Then how can we represent this peptide bond NHC double bond O? Okay.
Yes, ma'am.
You know it is cis or trans in nature? Trans in nature. Yes. Okay.
Yes, ma'am.
Then the alpha helix is stabilized by extensive hydrogen bonding. It is formed between hydrogen atom attached to peptide bond N and O atom attached to the peptide C. The hydrogen bonds are individually weak, but collectively they are strong enough to stabilize the helix. Here, there is not only a single hydrogen bond. We can see there are several hydrogen bonds. Yes, Vishnu?
Yes.
How many hydrogen bonds are there in this structure?
1, 2, 3, 4.
4. Okay, so simply it is weak, but it is collectively it is strong.
Yes,
okay. Okay, that's all. Then all the peptide bond except the first and last in the polypeptide chain participate in hydrogen bonding. Okay.
Yes.
Then each turn of alpha helix that contain 3.6 amino acids and travels a distance of 0.54 nanometer. The spacing of each amino acid is 0.15 nanometer. Like the structure of DNA, we discussed Watson and Crick model of DNA. Yes? You know the structure? Yes. There is a major groove, minor groove. Then we explain number of base sequences. Each turn of DNA that contain this much, right?
Yes,
like this, each turn of alpha helix contains 3.6 amino acids. Okay. Alpha helix is a stable conformation formed spontaneously with the lowest energy. Okay. The right-handed alpha helix is more stable than left-handed helix. So we can form this twisting in right-handed and also in left-handed. So the right-handed alpha helix is more stable than left-handed helix. Okay. If there is some amino acids like our proline,
but why? Why is the right one more stable? Because alpha helix is stable because of OH bond, right?
Yes, but in case of right-handed helix, it is difficult to attach the C double bond O and NH. When we study in detail the molecular level, we can see there are some difficulties. Okay, in left-handed helix. Okay, that's all. Then we are in this which point? Yes, 8th one. Certain amino acids like proline. Proline. What is the importance of proline?
Vitamin C.
Proline.
Is there
No.
a vitamin C, no?
Proline in vitamin C. What is proline? Proline is an amino acid.
Yes, ma'am.
Yes, it is present in vitamin C. Okay, functions of vitamin C. Okay.
Yeah, that's
Collagen
what I
synthesis.
learned yesterday.
Okay. Sorry. I just think proline is vitamin C. What is it? That's why. Sorry.
The
Proline.
hydroxy.
Very good. Hydroxylation reaction of proline and forms hydroxyproline, and vitamin C biochemical function is there. But what is proline? According to amino acids, what is proline? Huh?
It is an amino acid.
Importance of amino acid proline. In classification, you just note down which is the special group present in proline.
Proline is yeah, cyclic.
Cyclic ana. Then next proline that contain a special group. A special group is there. You find out like imidazole group in histidine, then indole group in tryptophan, and proline.
I don't know, ma'am.
It is known as an amino acid.
Yes, ma'am.
Okay. In all other cases, you just write down, it is known as an amino acid. Why it is known as amino acid? Because in all other amino acid, two functional groups like amino group and carboxyl group attached to the same carbon atom. Okay? Okay. Which are the two functional group of amino acids?
It is known as amino acid, right, ma'am? Amino acid.
IMINO. Amino acid. Okay. Why it is known as amino acid is that actually in case of all amino acid, NH2 and COH, both functional groups are attached to the same carbon atom. Okay.
Yes, ma'am.
You write it
Yes,
down.
ma'am.
That particular
Yes,
carbon atom is known as alpha carbon atom.
Yes,
Okay. But in case of amino acid, what happens? This NH2 group and COH are not attached to the same carbon atom. Okay. We have an additional point. Actually, this is coming under classification. We skip that classification. Okay. Only important questions are we discussed. That's why no problem. I will give you idea at the time of these classes. Okay. Don't worry. Okay. So you get the point. What is proline? Proline is an amino acid.
Amino acid, yeah. Because it is attached to different.
What is
because in every yeah.
You explain.
Alpha carbon is the carbon to which every other amino acids are attached to the same carbon. So that carbon is known as alpha carbon. But in proline, it's attached to different.
No, alpha carbon atom. You just consider the structure of amino acid. You can just draw the structure. We have seen the structure in our peptide bond. I will show you the diagram. Vishnu, you can see the diagram. Yes.
Yes, ma'am. Now, but I can see the sequence. Okay, now. Okay. Yes, ma'am. Yes, ma'am.
Yes. Okay.
Yes, ma'am.
Okay. Then you can see the structure of amino acid. First one.
Yes. Yes, ma'am.
C. This carbon atom is known as alpha carbon. Okay. The center C is known as alpha carbon.
Yes, ma'am.
Okay. To this carbon atom, amino group and carboxyl groups are attached. Okay.
Okay, ma'am. Okay, ma'am.
So in all amino acids, you write in a different way. That's why I take this picture. Okay. You are mistaken. Okay. Cut that written part. Okay. You just write it down. I will tell you that. Okay. The carbon atom to which amino group and carboxyl groups are attached is known as alpha carbon atom.
To which the amino group and carbon group are attached.
Not carbon, carboxyl group, COOH group, carboxyl group.
Yes, ma'am.
Okay.
Yes, ma'am.
Carbon and carboxyl both are different.
Yes, ma'am.
Okay.
I know.
Okay. So amino group and carboxyl group, these are the functional group of amino acid.
Yes,
Now
ma'am.
it is clear. So
Yes, ma'am.
these two groups are attached to a same carbon atom. Okay, that's why this carbon is known as alpha carbon atom. In all amino
Yes.
acid, there is alpha carbon, except which amino acid? Proline. Okay.
Yes, ma'am.
Okay, Vishnu. That's
Yes,
why
ma'am.
all amino acids are known as simply amino acid, but proline is known as amino acid. Hmm?
Yes, ma'am. Yes, ma'am.
Okay, this amino group and carboxyl groups are attached to different carbon atom.
Yes,
Okay,
ma'am.
in
Yes, ma'am.
case of proline. Okay,
Yes, ma'am.
that's why it is known as amino acid. Okay,